Hi, my name is Samara Rangel, and I'm going to talk about wildlife and the emergence of infectious diseases. As introduction, wildlife is considered a potential source for emerging infectious diseases that represent a threat for public health. However, the increased interactions between humans, domestic animals, and wildlife facilitate spillover of diseases. This spillover from domesticated animals and humans could be a threat to wildlife, whose populations then act as reservoirs and/or amplifiers of emerging diseases for domestic animals and humans. The effects of emerging diseases on wildlife populations are generally considered of less importance compared to other anthropogenic problems. Nonetheless, the understanding of the ecology of infectious diseases in wildlife and their impact on the dynamics and sustainability of these populations has been improving. This is particularly important for the recognition of the threat in the genetically impoverished endangered species and the significance of preserving biodiversity in order to prevent and control the emergence of diseases. The processes that are involved in the emergence of infectious diseases in wildlife include ecosystem alterations of anthropogenic or natural origin, movement of pathogens or vectors via human or natural agency, and changes in microbes or in the recognition of emerging pathogens due to advances in the techniques of epidemiology. So the factors responsible of the emergence of diseases in wildlife usually fall into more than one of these categories. In this presentation, an example of an emerging infectious disease that affects a particular wild species, and an example of an emerging infectious disease of wildlife origin, but occurring in Australia, will be given, explaining their history, epidemiology, impact on animals and people, as well as their programs of control and surveillance that have been implemented. The first disease is chytridiomycosis in frogs. Chytridiomycosis is an infection caused by the fungus Batrachochytrium dendrobatidis that has caused widespread population declines in both captive and wild amphibians of the orders Anura, which includes frogs and toads, Caudata, which includes salamanders and newts, and Gymnophiona, which includes caecilians. The disease can be found in Africa, the Americas, Europe, Australia, New Zealand, and Asia. History. The origin of chytridiomycosis is unknown, but a recent study proposes that it was originated in Africa, where it was first reported in the 1930s. However, Batrachochytrium dendrobatidis is a common agent in African frogs. African frogs that have been widely exported from Africa include the African cloud frog that has been exported since the 1930s for use in pregnancy diagnosis, as laboratory animal models, and as aquarium pets, and the dwarf American frog that has been disseminated in the aquarium pet trade. Therefore, the sudden appearance of chytridiomycosis is thought to be due to the international trade, which allowed the recent introduction of the agent into new regions of the world, and so the infection of new amphibian species. Chytridiomycosis in Australian frogs was first discovered in Queensland in 1993. However, the oldest record of P. dendrobatidis is from a museum frog specimen collected near Brisbane in 1978, which coincides with sudden frog declines in some species and the extinction of two of them in the region. Subsequent amphibian declines in central coastal Queensland around 1985 and the wet tropics between 1990 and 1995 suggest that the fungus spread north to its current northern limit at Big Tableland near Cooktown. In southern Australia, the spread of P. dendrobatidis is poorly documented, but its distribution extends down the entire east coast to Tasmania, where it was first detected in 2004. Two separate foci occur in other states, one in southwestern Australia, where the earliest record dates to 1985, and another around Adelaide in South Australia, detected in 1995. Chytridiomycosis is now found across Australia, except in the Northern Territory, as it appears to be confined to the cool and wet areas of the country. Only few areas that are suitable for the fungus remain uninfected, and some pockets of disease-free areas exist within infected regions due to the isolated nature of amphibian populations. Impact. The fungus invades the surface layers of the frog's skin, causing damage to the outer keratin layer. Amphibian skin is unique because it is physiologically active, allowing the skin to tightly regulate respiration, water, and electrolytes. It is not yet known exactly how the fungus kills amphibians, but it is thought that it may cause mortality through disrupting the normal function of the skin, resulting in electrolyte depletion and osmotic imbalance. In some frog populations, the disease causes 100% mortality, while only causing some deaths in other populations. Some amphibian species are highly susceptible and die quickly, while others seem to be less susceptible. With antifungal and supportive treatment, adult frogs and tadpoles can fully recover from the disease. In Australia, the fungus has been directly implicated in the extinction of at least 4 species, and the decline of at least 10 others, including Littoria nanotis, Littoria reucola, Littoria spenceri, and Nactimistes dayi. The 4 species listed as extinct are from Queensland and include Rheobatrachus, who was last seen in 1981, Rheobatrachus vitellinus, last seen in 1985, Traudactylus acutirostris, last seen in 1997, and Taudactylus diurnus, last seen in 1979. Epidemiology. Morbidity rate. When frogs show clinical signs, death usually follows within 2 to 3 days. Prevalence of infection in apparently clinical frogs in infected populations in Australia can approach 100%. Mortality rate. In captivity and in challenge and transmission experiments, some adult frogs are capable of surviving and clearing infections, but mortality rates of up to 100% are common. Pathogenicity varies with host species, fungal strain, exposure dose and period, temperature, and body size. High mortality or susceptibility to infection observed in the laboratory may not always occur in the wild and vice versa. Recently, metamorphosed frogs appear most sensitive to the disease in some species. Infections of tadpoles are limited to their keratinized mouthparts and often appear to have no negative effects, implicating them as potential disease reservoirs. Although some evidence suggests that infected tadpoles of some species may lose body condition and suffer reduced survival, tadpole infections can be carried through metamorphosis and cause high metamorph and juvenile mortality. Presence and prevalence of PD in the wild varies with species, life stage, season, altitude, and latitude, and may be broadly governed by temperature. It appears that amphibian populations develop some measure of resistance to the fungus over time, or the fungus becomes less pathogenic, or both, as frog populations have been identified that declined dramatically when the fungus first arrived, but now appear to have stabilized and coexist with the fungus. Studies have also shown that individual frogs develop and acquire immune response, allowing them to clear subsequent infections more rapidly. Incubation period. Time to clinical signs and death, usually ranging between 14 and 70 days post-exposure. Transmission is usually via zoospore and waterborne. Source of agent includes shedding of zoospores from infected skin. Zoospores leave the host via discharge papillae projecting through surface of epithelial cell. Zoospores require water to survive, although a film is adequate. Skin on frogs and mouthparts on tadpoles are the only infected tissues. Zoospore invades stratum corneum to infect new host. Frogs are subclinical for the majority of the duration of the infection. Frogs can be infected from water containing zoospores generated either from frogs or potentially from non-parasitic growth that might occur under certain conditions, though this has not been demonstrated to date. Chytridiomycosis is listed as a notifiable disease in Australia's national list of reportable diseases of aquatic animals and by the World Organization of Animal Health. In the Aquatic Animal Health Code. Control. There are no proven methods to control the disease in the wild to date. For amphibian species currently listed as endangered, emergency measures are needed to increase population sizes via reintroductions, translocation, and the establishment of captive disease-free assurance colonies. Since B. dendrobatidis is now widely distributed in Australia, control efforts should be aimed at protecting uninfected areas. As strains vary in virulence, reducing the risk of spread between infected areas is also important. Developing a greater understanding of how the impact of chytridiomycosis in infected wild populations can be better mitigated will help in controlling the threat of the disease. Surveillance. Monitoring and surveillance is necessary to monitor the impact of the disease on frog populations, detect new outbreaks in currently uninfected populations or locations of unknown disease status, establish restricted and controlled areas to which quarantine and movement restrictions are applied, establish infected and non-infected area zones, monitor the progress and success of a control strategy. Infection of amphibians with the amphibian chytrid fungus has been listed as a key threatening process in Australia by the Commonwealth Department of Environment and Heritage. Surveillance for chytridiomycosis, especially in disease-free areas, is proposed in a threat abandonment plan. This plan came into force on 7 September 2016 with two goals: to prevent amphibian populations or regions that are currently chytridiomycosis-free from becoming infected by preventing further spread of the amphibian chytrid within Australia, and to decrease the impact of infection with the amphibian chytrid fungus on populations that are currently infected. Risk analysis performed by Biosecurity Australia in quarantine requirements for the importation of amphibians or their eggs into zoological facilities did not list chytridiomycosis as a risk since it is endemic in Australia. However, this disregards the risk of importation into chytrid-free areas. Although chytridiomycosis is not specifically mentioned, the general hygiene strategies recommended will prevent the release of imported strains of B. dendrobatidis during the initial 2 years. After 2 years, the amphibians can be released without testing for B. dendrobatidis. However, if being released into a chytrid-free area, the same requirements imposed on Australian-bred amphibians under the threat abandonment plan will apply. Amphibian chytridiomycosis was listed on the OIE Wildlife Diseases list and has been declared an international notifiable disease. Australia now reports to OIE on its chytrid status via Aquatic Animal Health Committee.